My name is Nicholas Hanle, and today I'm going to review with you my concept development project on stem cells and their use in medical research. The objectives of my presentation are number one, to define the properties of a stem cell, two, to compare and contrast the major types of stem cells, three, to discuss the current use of stem cells in medical research, four, to identify future uses of stem cells in medical research, and finally discuss the ethical aspect of stem cell usage. So we're going to start next with the importance of stem cells. Now, according to the National Institutes of Health 2015, stem cells are cells that are undifferentiated. This means they have the potential to develop into multiple cell types. What's unique about them is when a stem cell divides into two daughter cells, each daughter cell can choose to either remain a stem cell or they can begin the process of differentiating into specialized cell types. Stem cells have the capability to renew themselves through cell division, and the time waited between cell division may be as short as a day or as long as years at a time. So, things like brain cells and heart cells tend to have a longer time between their cell divisions, whereas things like the stomach lining and everything that's replaced pretty often has a shorter time period. Um, Stem cells can also be induced to differentiate under certain physiological and experimental conditions. So, by studying how these cells differentiate, it can help us with understanding of how embryonic development occurs and, in reality, how cancer develops by watching the cell cycle and seeing what proteins and other cell signals are available. Cancer development can be processed there. Differentiation can be induced through internal cellular signals, such as through the expression or inexpression of proteins and genes, but it can also be regulated through external signals, such as signal molecules from other cells, cell to cell contact, or other microenvironment molecules. Overall, stem cells have the potential to treat many diseases and disorders, such as heart disease and diabetes, as well as degenerative conditions such as um, Huntington's disease and even Alzheimer's. Stem cells could hold the keys to regenerative abilities to help with the healing the body from diseases, injuries, and disorders. The field of regenerative or reparative medicine deals with this. Um, Again, like we said, diabetes, heart disease. If we know the beta cells in the pancreas aren't working, we can develop stem cells to kind of induce those or mimic them and create them as we go. Okay, now the types of stem cells. There are three major types of stem cells according to the International Society for Stem Cell Research. They define the types as tissue specific stem cells, embryonic stem cells, and finally induced pluripotent stem cells. Um, Tissue specific stem cells may also be called adult or somatic stem cells. These stem cells are multipotent, which means they can't become just about anything, but they can become anything on a particular cell line. Um, for example, bone marrow stem cells may be able to give rise to bone marrow, red blood cells, white blood cells, and other cells along that same cell family. Um, Adult stem cells are found in many organs and help the various organs replenish lost or damaged cells, and there are often very few of these in each organ, making isolation of them difficult at times. Embryonic stem cells are stem cells taken from early embryonic tissue. These are found in, like I said, the blastocyte of the embryo. Most of the time, these cells are gathered from in vitro fertilization, fertility treatments. The embryos that aren't used get disposed of, and they use the research for that. But these cells can also be gathered through miscarriage or abortions, which makes this one a little bit more of an ethical issue of how to gather these or not. And we'll talk more about that in a bit. Um, Embryonic stem cells are pluripotent. so they can do multiple, pretty much any cell in the body because they haven't decided. They're in that initial 100 to 150 cells in the blastocyst, so none of the organs or anything have developed yet. So embryonic stem cells have that potential to develop into whatever is there. Um, and then the final type, the induced pluripotent stem cells. These stem cells are kind of reverse engineered. Um, they're created through basically adult cells, such as skin cells or another thing, can be reprogrammed to become pluripotent, like an embryonic stem cell. This is done by adding specific genes that are necessary to turn embryonic stem cells into specialized cells. So they kind of reverse this. These are going to be pretty important. We can study diseases with them. They're good, and we'll talk about them in a little bit. Okay, methods I would use to introduce stem cells to a class of AP students. An activity I've done in the past has been a jigsaw activity. Basically, I divide the class into groups of three if I have three cell types. So groups of three, and each member of that group is going to be inside one of those cell types. Those members are then each assigned to become an expert on their assigned cell type. So there would be a research project they have to do with it, or they could go through and I'd have a sheet with the main requirements that I need them to find. They would have to go through and determine what the important details are for their particular cell type. And then once they feel that they're the expert, they completed the sheet that they need to, they would go back and then teach the other members of their group about their particular stem cell type. It's helpful to have, like I said, a data table of list of things that they can require so that they can make sure they have all the necessary material. I've done this in the classes. It gives a good way to introduce the activity. They kind of have an idea what stem cells are, but this gives them a way to see the different pieces without having to research a whole bunch of stuff. A second thing I could do, and like I said, number one's kind of my go-to, but number two, an independent research assignment. On this, there's so many different things about stem cells that they could discover. Letting them go and either pick their own topic or have a list of topics where they can pull from a hat and just develop a short, brief research assignment on. It could be a paper, it could be a PowerPoint that they make. They have some options here of how they can present it, but anything where they're independently looking stuff up, and hopefully we can find something that's of an interest to them. The final way is an ethics discussion, particularly with embryonic stem cells and the way they're collected or obtained or potentially obtained. It could kind of stir up some hot feelings around the classroom. So getting into that and letting the students debate how they feel and how they see things. They love controversy, so this is a way for them to see, and you could even do a debate on this where you set up half the class is for using them from any source and half the class is against, and give them a chance to look up arguments for and against and then have that little discussion. It just kind of hopefully ties them in and debate whether or not that the obtainment of those cells is ethical or not. Okay, so some critical information, details, and examples. Basically, what I did here is through the research, anything that kind of stood out to me as something that I felt was important, I'm going to kind of talk about here. So, the first study that I came upon was actually two people or two groups. Colpo, Fur, and Stymie, and Chichera in 2019, and Mahula and et al., a group of people. They looked at mesenchymal, neural, and induced pluripotent stem cells to see what effect they have on the treatment of Alzheimer's disease. And they were using animal models, but just to see the treatment there. And what they found is that all of these cell types showed promise in treating the condition. Mesenchymal stem cells were the most commonly used, and they worked mostly through indirect methods. Basically, you insert the mesenchymal cells into a target region, and with the cell signals and stuff that they excrete, they are able to induce the cells around them to regenerate, repair. The The neural stem cells have shown they can differentiate into neurons, astrocytes, and oligodendrocytes. And then the induced pluripotent stem cells have shown success similar to the neural stem cells. Again, all of these have been shown in animal models, so it's not kind of ready for human testing yet, but we have some positive things here. The second study was that Varus Godi and al. Found that menstrual stem cells or mesenchymal stem cells from menstrual material can be analyzed to determine if a female is at greater risk for preeclampsia during pregnancy. Basically, they found that in patients with preeclampsia, the stem cell, the compounds they were being produced are different if you have preeclampsia or if you don't. So by analyzing these, they found that women with preeclampsia showed um, reduced angiogenic capacity and reduced stromal progenitor cell formation when compared to patients without preeclampsia. So angiogenic capacity, obviously the placenta is supposed to, tie the blood in between the mother and the child. So if we're not developing that, the implantation makes it a little harder, which could explain some of the symptoms or things that evolve with preeclampsia. Um, Preeclampsia patients' menstrual stem cells express less endoglin and had an increase in pro-inflammatory factors. It's important because it can be used to develop predictive models to scan for patients that may be predisposed to preeclampsia. And then further study of the menstrual stem cells could also lead to a greater understanding of how and why preeclampsia occurs. So that was from Varus Godat et al. In 2019. Travanikova and Bakakova in 2018, they studied adult mesenchymal stem cells, and they used them in bone and vascular tissue engineering. And what they found is that they could be used to engineer tissues to help repair damage to bone and blood vessels. So bone marrow stem cells can be used to regenerate cartilage in the knee because of osteoarthritis, to help fractures. They can help fractures heal more uniformly, and they can interact with scaffolding material to also repair bones. So they have a lot of promise there. And then adipose-derived stem cells can be easily collected via liposuction. And then once you collect them, they're being used currently in cell-assisted lip transfer, lipotransfer, or CAL, a form of plastic surgery used to fill in and augment various parts of the body. These cells can also be used to accelerate the healing of diabetic wounds or in the treatment of chronic limb defects. Other areas where adipose-derived stem cells have shown promise are in hair regeneration, peripheral nerve repair, cardiovascular repair, amongst other things. So the adipose-derived stem cells are showing a lot of promise for future investigation. Al-Karim et al. Conducted research in which neuroectodermal stem cells were used to regenerate the fine structure and locomotor function in rats that had been injured with compressed injuries to the spinal cord. Basically here, before the stem cells were implanted, they had the group that didn't have the neuroectodermal stem cells implanted, and then they had a group that did. The spinal cord injury caused the loss and degeneration of many nerve fibers, causing the myelin to split and the mitochondria to degenerate. But with the insertion of the neuroectodermal stem cells in the one group, they were able to remyelinate in significant numbers central and cranial to the injury site, and they were able to increase the locomotor function both in frequency and in duration. So there were a lot more movements, and the movements were for a longer bit of time. And then finally, Mansur, went too far. Mansur et al. In 2018 were able to develop insulin-producing cells from human-induced pluripotent stem cells, but they had to use a scaffold to do this. When they initially started this one, they tried to grow the cells in culture. The cells in culture would not form the three-dimensional structure necessary in order to mimic the beta cells of the pancreas and the islet formation, so they weren't able to produce the insulin properly. But by using the polyether sulfone or PES nanofiber scaffold, it was able to allow the cells to form the 3D structure necessary to form an islet, and they actually produced insulin for the or functioning human insulin. So it's showing promise there as well. Okay, for my expansion of the topic, there's two things that I came up with to kind of, if my students are getting on, we can expand them further. First is Bunnell et al. In 2008. Their research that I read explained in detail, and I mean in detail, how to isolate, expand, and differentiate adipose-derived stem cells. It went into the specific solutions you needed, and with differentiation, trying to get cells to do that, those mixtures are very important. Um, what I could do with this is my students could explore that detailed pathways, the materials, equipment necessary to work with the stem cells, give them that big initial or advanced foundation in that. Um, the other one is I come from a pretty conservative area of the country here in South Carolina, and ethical debates can get kind of one-sided a lot. So the article I found from Kurzer in 2004 provided, I'm calling it an interesting take on the ethics of embryonic stem cell research. Um, his viewpoint is definitely not conservative. So having the students read through his paper and seeing what his arguments want kind of provides them with that other side of the story that they generally don't see, or maybe not have ever thought about. So trying to give them that opposite side of that could allow them to expand on that and provide that second viewpoint for them to work with. That concludes my presentation. Thank you for listening.